Hi everyone, I'm Kai.
What does evolution mean?
That means they get bigger and stronger, and some of them can also learn new moves.
Okay, say bye.
Bye!
So when I asked my son what evolution is, you see how he responded. Okay, in his mind, evolution is really equate to Pokemon, and I'm not gonna lie, I grew up with Pokemon, so I think a lot of you also quite fond of it. Not all, but I did saw some of you sometimes play Pokemon Go. Okay, and if you saw me use my phone sometimes in class, you may all have also noticed my phone case has a Pikachu things on the back. So anyway, enough with this small talk, and let's get to the topic of today. Now the title here is Directed Evolution of Nucleic Acids, Molecular Recognition Elements. So the focus of Tuesday was on amino acids, right? So basically we have peptides, enzymes, antibodies. They are all protein or amino acid is the building block. And this time we're talking about nucleic acids, okay? Now this is maybe something a little bit new for you, particularly those that have never really talked to me in regards to research. Those that have been doing research with me, you guys know, you know. So that would be a little bit revisit, a little bit fill in of the informations. And I'll also, with the subtitles of past, present, and future. So looking at some historical perspective of the field, what's going on right now, in particularly what I have been doing with students, or more like in my recent past, okay, and also future, okay, what is the outlook of this field, in particular, can we tie it into COVID-19 or SARS-CoV-2? Alright, so with that enough said, again, this video is for a whole bunch of audience, okay, for the P2s, okay, that's the majority of audience, for several P4 that are doing a elective AP activity with me, and as well as for a bioengineering student in the MS program. Alright, now this lecture, I wouldn't say lecture again, this is more of a seminar presentation, so I try to think of it like that. I hope everyone would pay attention to this seminar lecture. Stay awake. I know sometimes my voice goes up and down, and when I get excited I got louder, when I got tired or something I got softer, so try to stay awake, and watch the video in entirety, okay? There's a very good reason to watch it for the entirety, and you probably don't really need to take any particular notes on these things, okay? And don't worry about how this material will be tested in the final exam, okay? Don't worry about final exam. I posted an announcement saying that I will put up a second announcement on Friday, and actually that announcement, it's scheduled to release on Friday, so I try to wait it until Friday, so don't worry too much, okay? I don't know what else I can say. It would be very, 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 very reasonable. Alright, so and then the lecture outline, or the seminar outline, is in the next 2 slides, okay? And with that said, let's have a little bit recap. And previously on Dr. Han's biotechnology lecture. Alright, and then we talked about SARS-CoV-2, right? And some of the highlight, we spent about half of the last online lecture or video lecture on the SB domains, how it orients, how it opens, and it's really we believe right now is the key thing for it to bind. It's like the hook, right? To bind ACE2 of the target cells, and the lung cells, alveoli cells, one of those, and many other cells or tissues also express ACE2, and that's one of the reason it leads to some of the systematic immunological flare and lead to very deadly situations, okay? And we visited those ideas directed evolution of enzymes, okay? And I propose, hey, is it possible to evolve a ACE2 variant as a decoy for the virus? Maybe it will work, okay? Maybe it will work. Someone that is directly in the enzyme evolution field, I'm sure they are looking something at that, in that extent. And phage display library, okay, of peptide and antibodies. So can we select a antibodies, okay, rather than using hybridoma technology, rather than using a mice to do the job? Can we do a in vitro selections, okay, using cell, basically animal-free system to produce what we need to bind to, maybe in particular the SP domain. So that's from Tuesday, okay, that's from Tuesday, and we kind of wrapped up the amino acid side of the story, and let's go down to more of the basic building block, right? We always talked about the central dogma from DNA to protein, to expressions, right? Things like that. Now we move one step back, and in this episode, okay, in this week's, in this today's, topic, okay, we are going to look at the biomolecules that what are the biomolecules can capable of binding, okay, that is not amino acids. Well, basically the title tells you something based on nucleic acids, okay. The past, okay, what are these biomolecules, okay, the discovery, the properties, and why are these molecules so special, okay. Looking at some of the diagnostic therapeutics in biosensing type of a thing that I've done in a little bit more past or recent past. I consider those as present, okay. Anything within a 6 years, it's present to me. And then what about the future? And basically you're looking at my point of view, okay. What can researchers in this field, a particular field, can do or in to combat SARS-CoV-2, what we can do. Okay, now, so that is the preview of this episode, and let's dive down into the meat. Alright, so we'll first talk about the past, okay? The past, we're talking about the pioneers of the field, okay? Now, let's fill you in with some of the basic terms and whatever is going on. Okay, a lot of the time when we're talking about something that can bind to a target, okay, like last time we talked about peptides, enzymes, or even antibody fragments, okay, they can bind to a target, okay, and target in this sense, can be in proteins or small molecules or whole cell bacteria, okay, it could be many things, okay. Now these binding elements, collectively we could call it molecular recognition elements, okay. Basically they can recognize another molecule and in a specific tight way of fitting, and usually these binding, we say it as bind with a high affinity and specificity, and really that is the basic criteria for something to recognize an element. If you don't bind with high affinity and specificity, it's pretty useless, okay. It's not good enough for research or any applications possible. And now we talked about peptides and amino acid, and this term also include nucleic acids based molecular recognition elements, okay. And what are the nucleic acids that I'm referring about? Yes, okay, DNA, okay, DNA. Okay, now in many cases we're talking about single-stranded DNA, one piece of DNA, or RNA. Okay, RNA. Now RNA is single piece, one piece by nature, so we don't need to put ssRNA there. Okay. All right. So these particular molecules in the nucleic acids molecules in very particular way, there is a name called aptamer. Okay, aptamer. Okay, so that is the title name. So what does aptamer mean? So this term comes from a Latin aptus, means fit, and Greek mer, okay, like anything more, okay, polymer, all these things, fit part, something that fit, okay. And like I said, it could be oligonucleotide, peptide molecules that bind to a specific target of molecules, and these aptamer are identified through a screening process, of so-called like in vitro selection. So it is a process that is virtually almost the same as we described for the peptide phage library screening. It's an iterate process. You look at binding, wash, and take it out. So we look at it again, okay, but it is a in vitro selection process, i.e. no cells basically is involved in the process. And this particular term aptamer was first described by Dr. Andrew Ellington and Dr. Jack W. Szostak in 1990. And remember in 1990 from last time when I talked about phage display library, and it was published in 1990, and so many things happened in 1990. This particular article was published in Nature and other big scientific journals, and almost everybody wants to publish in Science and Nature. It's almost like the second best thing after of getting a Nobel Prize. So in that experiment, they look for RNA molecules bind to a specific ligand. This we have a famous people again, Jack Szostak. Okay, he actually is the 2009 Nobel Prize laureate in physiology or medicines. Okay, now last time we have chemistry Nobel laureate in 2018. This one is even earlier, Dr. Szostak, and his credit was actually not really directly related to these aptamer research, but actually a step backward into what he was doing is actually the origins of life. Isn't that cool? The origins of life. How or why does life exist on Earth? Alright, so that is the biggest questions, other than, are there any other creatures out there in the space, right, in the universe? I think that is a cool, very cool origins of life. And actually NASA, NASA, Space Foundation or States, whatever, they have a particular research grant in the category called the origins of life. And locally, I would say locally, in the state of Pennsylvania, who I'm aware, there is a researcher in the Penn State State College. There is a researcher that do these type of origins of life research also. Okay, so that is a little trivia note. Alright, so what in their work, what did they do? They attempt to answer the questions of origins of life and early evolutions of life, really. So they find out these aptamer thing really was by accident. They were doing research just to trying to look for origins of life, and somehow it's a byproduct, almost like a byproduct in a way. So they in those in this origin of life theory, okay, a little bit of it presumably, they were thinking the first biological catalyst arose from a random sequence of polymers. It could be like polynucleotide or peptide chains. Okay, it's just random. Okay, here is a picture that if you search primordial soup or something and then on Google, and you'll find something. So initially there was some type of organic molecule, okay, small organic molecule, and some random chances they kind of stick together, and really, the earliest form we're looking at some type of a nucleic acids. Okay. Now, forming these random sequence, it's just random, and chances of these random sequence to form a stable three-dimensional structure that have binding and catalytic activity, i.e. the basis of life, okay, it's believed to be very, very low. It's not that easy to have life, really. Like if you put a soup of organic molecule in a beaker, set it on the bench, chances are it will probably take billions and billions of years for something to happen spontaneously, or it may not even happen at all. So it is very, very low chance. And then, so in order to study these low chances of events, they try to recreate these evolutionary type of a scenario. Okay, these nucleic acid evolutionary scenario in a lab. Okay, now they need to do it fast. No one has billions years of life, okay, to live to watch these things happen. So they designed a sequence of pool, okay, a random pool with RNA molecules, i.e. the A, okay, U, okay, G, C, AUGC, right? Not ATGC, because this is RNA. And it's all random. Now, don't worry about the sequence, the details, how long it is. Don't worry about it. It's just a random pool, and then they believe to have a 10 to the 13th different molecules. So they have enough combinations, okay, to create these differences. Okay, the idea here is if you have 4 code here, if you have 4 positions, okay, and basically you do the probability of permutations and things like that, you have 4 to the 4th different combinations, right? You have A, U, G, C, C, and then you can mix and match, and basically you have this much of a variant. Now, if you have a long enough chain, you can create a lot more variations, and usually it's up to about 10 to the 13th. How many zeros? I don't know, but it's just 10 to the 13th. Alright, so from their experiments, okay, they isolated the RNA sequences that bind to a particular target. Now, in this experiment, they suggest or suspected, the chances, okay, the chances for here is a typo here, sorry. The chances for isolating novel ribozymes from a random pool, it's not 0, okay, because of this experiment. They were able to speed up this process, and what ribosome is, they are the catalytic RNA, okay, that, some of the early form of catalytic biomolecules, okay, i.e. tying back to the origins of life, okay. Now, RNA here, in terms of what you are familiar with, we have the mRNA, right? Messenger RNA that code for protein. We have tRNA that charge the, bring the amino acid in during the synthesis, and we also have rRNA or ribosomal RNA. Now, those RNA itself have some catalytic activities alone. Okay, so that tied into the origin of life work, and they were saying RNA world hypothesis, suggest that, these, free-floating nucleotides, are the earliest biological catalytic functions on Earth or elsewhere. Okay, it could be happening elsewhere, and really that is the the earliest form of living thing, you could call it. They are not really a living thing, but anyway, so people believe we have RNA first, and then slowly evolve into DNA, and somehow the DNA kind of took over for majority of the living things in the world. We have RNA living organisms, right? Our beloved or hated, SARS-CoV-2 is an RNA viruses. Okay, so again, those are long history, but that it's almost looked like this aptamer discovery was somewhat an accident by Ellington and Szostak. But there's a big but in science. It's always happened about the same time. There is another group of people called Dr. Craig Turk and Gold. Okay, Dr. Larry Gold. Here we have Dr. Turk here, and then Dr. Larry Gold in the gray hair here. Now, Dr. Larry Gold is a very high profile people actually in the field. He's speaking everywhere, but Dr. Turk is very low profile. I've never see him presenting anything in the public actually for any years for all the years that I've been in the field. Now, they published in Science. Look at the day, 1990, August 3rd. Okay, now when they published in August 3rd in Science, they probably started some of the research back in 1989, and so what they performed a similar experiment as compared to Ellington and Szostak, and they selected again RNA ligands to a polymerase, DNA polymerase, is a bacterial phage T4 polymerase. Okay, so they didn't give the name for aptamer, but they give the name for the process, the in vitro selection process. Now notice Zostech, they all just call it in vitro selections. Okay, it's very boring name, but they find out a new name. It's called systematic evolutions of ligand by exponential enrichment. A mouthful of term, and usually people abbreviate it as SELEX. Okay, taking some of the heading there to as a SELEX. Now, because they started the process a little bit earlier than Ellington and Zostech, and that what happened is they patented. Okay, Larry Gold would have very good business mind. Actually, they patented the the commercializations of aptamer. Sorry, again, there is this typo there. I will fix it in the post that I give it to you. And the related process and products for 20 years. All right, for 20 years they have a monopoly basically. Now people can do research in the academic world, but you cannot sell these things. Okay, because Dr. Larry Gold, he was ahead of everyone. They also lead to founded the pharmaceutical company called Nextstart based in Boulder, Colorado, and lead to the approval of the first and actually only aptamer drug, Macugens, or pegatinib. Okay, that is indicated for treatment of age-related macular degenerations, but that's back in about 2005 to 2006, about that time. So it is more than a decade, right? So like any other drug developments, since the, bench research to marketing, it's more than a decade of time at the very least. Okay, now remember this Macugen I talked to you guys in self-care, right? In the eye topics, right? So it's a couple months ago. It seems to be a very long time ago. I know time seems to be kind of weird feeling right now. Alright, so Larry Gold also founded another company called SomaLogic, also based in Boulder, Colorado. Okay. So just some of the trivial FYI thing. So you see that one group, okay, went on, okay, to one of the person at least, okay, Dr. Zostech, okay, got the Nobel Prize, okay, got the prestige. Alright, now unfortunately Dr. Ellington didn't get anything. He was a postdoc at the time when he published this article with Dr. Zostech. So he didn't get anything, but he did get credited for this aptamer discovery a lot of time. But another group went for the industrial route. Okay, they get the money. Okay, they get the money. That definitely very lucrative for a very long time. Now, right now there are more small companies start popping up doing commercializations of these aptamer product here. And you ask me, can Dr. Hong commercialize what he's doing? Now, yeah, I do aptamer, just in case you're still not sure what I was doing. Yeah, theoretically I could, but I don't have the what should I say, the motivation yet. Yeah, motivation. Money doesn't motivate me enough yet. Okay, so that is the long story short. Okay, now so what is this systematic evolution of ligand by exponential enrichment? Okay, a mouthful of a process. Okay, so it is again a repetitive cycle, very similar to what I presented to you in the peptide display library and evolution of enzymes. So evolution is just keep going on and going on, generation after generations. And here we use, instead of saying generation, we just say rounds and rounds of this selection cycle. Now again, you have a target, you have a library. Okay, you look for something that binds, they will amplify it, and something that doesn't bind will wash away. Exactly the same as the phage display library figure that I presented to you, right? Now there's some extra step you can add on to it. Now you want something that bind, you retain. If you introduce something called negative target, or something that you don't want, undesired target, okay, here, and you can screen for things that do not bind, and then you keep them and amplify, and again repeating the cycle. So over the time, your pool will start narrowing down, having more of the populations that are capable of binding to your specific target of interest. Now these experiments takes longer. Nucleic acid they are more difficult actually to form good structure with good affinity compared to peptides and proteins and antibodies. So when we talked about peptide screening, we're talking about maybe 5-6 times usually is enough, but for nucleic acid it could take up to 10-12 times or even more depending on what you are screening for, what the target is. So it's a little bit longer process, and that's what we do in the lab here. Okay, now there is a anecdote story. Okay, there has been a rivalry between Dr. Andrew Ellington and Dr. Larry Gold. Now, Dr. Szostak, he got a prize, right? He knew he's going to get a prize, really. He's a very prominent figure. He doesn't care about money at the time, and he was in Harvard, and I think he was in Harvard at the time. So it really, he doesn't need that type of money. Larry Gold and Turk as well. Gold and Turk, they founded the company. Okay, they start making money. And what about Dr. Ellington? Well, he ended up having he's still at Texas A&M actually, and he have an academic career. Okay, but he gets almost nothing. He couldn't make money. He didn't get the real fame. So there's an anecdote story about phone conversations between them, and I'll tell you where I hear that later. Okay, I'll tell you where I hear that. It's a phone conversations on. What I hear it's on the Dr. Andrew Ellington side. Okay, he was on the phone talking to Dr. Gold. Okay, supposedly this is Gold. Okay, this is Ellington, and Dr. Ellington was on the phone yelling, "I'm gonna kill you. Okay, I'm gonna kill you." He was so mad on the phone yelling to Dr. Gold, and I don't know the details of that conversation. Neither does the person that told me know the detail. But the person that told me, saying that Dr. Ellington was very, very mad, angry on the phone with Dr. Gold. You figure. So where did I hear that? Stay tuned to the end, and we will get to the end of the story. Okay. All right, so that is the historical perspective, and then let's move on to some of the present thing. Now, it is my perspective. Okay, the field is 30 years old. Okay, if you count the first published 2 paper, it's coming to its 30th birthday. In a couple months. Okay, in the past 30 years, there's been a lot of group doing these type of research, relatively a lot, but still a relatively small field. Okay, a large and a small field anyway. So locally, in the northeast PA, basically it's just me. If you go a little bit further in the radius, there are a couple groups in New York City, one in the Bronx area, one in the Manhattan area, and the group in the Manhattan area, Dr. Ryan Williams, actually, he was my lab mate during graduate school. So we ended up both having a faculty position. So it's kind of rare actually for the 2 person in the lab actually have this opportunity. And further down, Florida, there are some prominent figures down there, and there are some international group, someone in Canada, McGill University, there are some, and going further, Korea, there are some very prominent figures, and as well as some groups in the Europe. Okay, now so that's just pure. So you see there are some people out there. Okay, it's not too little, but it is little enough that for me that has not been in the field for that long of a time. Okay, that I can be aware of who they are and where they are. So it is still relatively small. So we're just talking about my perspective here. Okay, and I'm not like the gold standard. Okay, I'm just one of the very early researcher in this field. Okay, just a disclaimer, not to be like just self-indulgent. Okay, don't take it as a self-indulgent sense that hey, Dr. Hong just talked about himself. But no, that is not the purpose. Actually, I'm talking about students' work more than myself. Okay. All right, so we are going to talk about a DNA application basically. Now it started with RNA. Okay, it started with RNA discovery, aptamer discovery, and people migrated to DNA the aptamer study, but in particular single-stranded DNA aptamer study. Now, one of the reason is just the inherent nature between RNA and DNA. Now, DNA is a lot more stable than RNA, and in terms of applications, DNA are much versatile. Okay, you can do a lot of manipulation with DNA, and they'll be fine. Okay, they'll be fine. So that is the transitions from RNA to DNA. In particularly some of the applications that are outside of the cell, so DNA are more favorable in many cases. Okay, so what are these things look like? Okay, so when we talk about antibody, we talk about a Y shape. Okay, how about these aptamer? They have many different shapes. Okay, could be many different shapes. Now, there are not a lot of 3D type of structure. Here is one example showing an aptamer. Okay, it still have somewhat of a double helixes that people describe, and somehow these things will twist. Okay, forming some type of a hairpin or loop stem structure by itself, even in a single strand fashion. So they are forming internal double bond, double strand. Okay, you can think of it like that. And these formations create some type of a pocket, and these pockets can interact with your target. And here we have an example of aptamer interacting with thrombins. Okay, thrombins, factor 2A. Okay, one of the clotting factors. And in terms of this is 3D and 2Ds, we usually just use these loop and bulges figures to describe its look. Okay, so it's kind of arbitrary, those are all just predictions. They may look like very different in reality. We don't know for sure. Now, what is the attractiveness of using DNA molecule other than its structural stability? Is the easy to reproduce them, right? So you remember the process have amplification process and amplifications to replicate DNA. What is the most easiest way? We learned that in couple weeks ago. Okay, we learned the PCR, right? Polymerase chain reaction. That is the easiest way to replicate these DNA molecule, to amplify them cycle by cycle, to enrich the pool, so that they are enriching to binding to the specific target. All right, that's one of the reason. That is the main reason that our lab is doing so many polymerase chain reactions all the time when we have active project running. So now you know what happened. Okay, that I told you a couple weeks ago. We use so much polymerase that we need to produce them by ourselves. Okay, that is the end. That is the finally telling you the answer. Why is that? And now, in terms of random region, I talked about it. Really, we're looking at 4 to the nth. Okay, 4 to the nth, and the n really equals to the length. Okay, of this random region. Okay, the length of this random region. So, for example, if this random region is 34, okay, nucleotide longs, okay, ATGC, or repeat in any random fashions, you have 4 to the 32nd diversity in theory. Okay, theoretical diversity, and they can all fold into this tertiary structure that we just talked about. Now, these things usually designed with some type of a constant regions, so that you can put a primer in. Okay, to do polymerase chain reaction. Think back a little bit of what we talked about couple weeks ago. So you need some type of a starting place, right? So those are some of the constant place to start, and inside usually is the randomness that is being given, the randomness in the middle. Alright, so that is in a nutshell of these DNA library, and you can just imagine there are just tons and tons of these structure. Now, not every structure have structure, not every sequence have structures. Okay, remember the chances. Okay, the origins of life. Okay, theory. We're saying that the chances for these random sequences to have a particular structure that can bind to something is pretty low. It's very low, actually. So there aren't that many. Okay, and if you want something that is good, even binding not enough, binding good, you need to go through these select process multiple times to help this pool of the soup of molecule to evolve in a way. Okay, so what are the applications? Okay, they can do research application, diagnostic, biosensor, therapeutics, drug discovery, biomarker discovery, synthetic biology. Pretty versatile. Okay, over the past 30 years, there's a lot of different aptamers being selected, and they've been really investigated in all these places. Okay, in particular, okay, we are going to highlight some of those and to talk about what has been done in a way, not necessarily all of my work, okay, but just trying to highlight the key points here. Now, therapeutics. So let's look at therapeutic first. Now, I'm sorry that it's not in a particular order from 1 to 1, but let's look at therapeutic first. And here is in 2002, okay, Dr. Ellington with his student at the time, Dr. Lisa Suter, okay, having published an article talking about novel therapeutic candidates in various fields. Okay, 2002 seems very long time for you guys. For me, it wasn't that long ago. It feel like, okay. Anyway, so more recently here we're talking about 2019, last year. Alex Pons, okay, graduate from last year class, okay, pharmacy class, Alex Pons, and he helped me wrote an article really looking at some of the clinical developments and potentials for aptamers for thrombotic events treatment and monitoring. Okay, so he wrote vast majority of the article. I kind of just chip in, do a little bit edit there, and a little bit end conclusions or perspective view. So he really did a good job at the time. That's why I'm saying this is a time to promote students. The time to promote students. Okay. Now here is the first student that I'm promoting. He's great. Alright, so therapeutics. Okay, it is an emergent area really. And what I've been looking at in the past year or two was to looking at some of the literature out there in terms of using aptamer for thrombotic event treatment and monitoring. Now, a lot of the DNA or RNA aptamers have been selected in the field that are targeting different factors involved in the clotting cascade or coagulation response and the clotting cascade there. Now, here are just some of the figure I extracted out from that particular paper. Now the P4s are going to read this paper. P2s and everyone else, I'm going to spare you for the requirement, but you are welcome to search it on the PubMed and go down and read it. Now I know you guys haven't go through the P2s haven't go through the cardio module, and you may not be as familiar. You may have forgotten the blood chapter that I taught you last year in P1, right? Anyway, you don't remember the clotting cascade. That's okay. It's not gonna be on exam. and not testing your physiology and things like that. But all the information is in that article if you wish to read it at your free time. Now, so these aptamers targeting different factors, the von Willebrand factor, VWF, factor 2 or activated factor 2, okay, thrombin. Again, I apologize for this typo. I don't know what happened with my typing here. It's factor IX, okay, I know what happened because S is about X. Now, again, I will correct all these typing errors in the version that you are reading, okay, in the version that are reading on screen, okay, or whatever way you look at it, okay. So all the factor that are activated, okay, all these involving factor 9, 10, all these have aptamers specific for it. Now, there are a lot of those, in experimental phase, preclinical. Some did go a little bit further, okay, in particular, so-called this REG1, okay. It is a RNA aptamer that target factor 9A. It actually go as far as to phase 3. Yeah, it go almost to approval, phase 3. It's efficient in many cases, but it failed at the end in a large trial due to, a large population due to anti-pegylation antibodies. Remember when I talked about it back in the GEM module this year? Okay, I know people never heard of that maybe. So pegylations, okay, pegylation is the common way to increase the durations, right? Increase the half-life of the biomolecule inside the body so that you don't need to do do like daily dosings. Okay, you can do weekly dosings perhaps. And these pegylations on the aptamer, okay, what generated antibody in the patients and render these molecule useless, render this reg1 reg1 whatever we call it molecule become useless. So that is the end of the story. It's very sad. It was almost out there, but it's stopped by these antibody. Now the sequence itself, the RNA sequence itself, were very non-immunogenic in nature. So this is the main advantage of nucleic acid binding elements compared to amino acid. Okay, they are very non-immunogenic in nature. Alright, so that's the table from the paper that summarizes the very current event, past and current, of what went on with different aptamers targeting different factors and where are they happening. A lot of them, they're kind of mixed between some academic or industrial academic. Okay, it started from academia and then a company got established from that. Okay, Duke Medical Center, they're very big on this anti-thrombotic aptamer development, so probably the leading group there. Okay. Alright, so what other therapeutic applications there is? So there is the cancers, okay, cancer treatments and infectious disease. Okay, those are all potential uses for these when we're talking about targeting. So the idea is the same for almost the same as those antibody drug conjugate that we learned about last week, right? So the antibody have a target, they carry a payload to kill the cancers and infectious disease, as some of you suggested in your assignments. And yeah, this is the same. Okay, aptamers is essentially could be replacing antibody in the future if things go into the right direction. So there are some company in the online, in the website, aptamer company saying that forget aptamer, forget antibody, use aptamer. So even with those slogans, I find it a little bit exaggerated at this point in time. Alright, so cancers, okay, what could be potentially happening here is an example that I drew 5 years ago, a long time ago, when I was presenting my interview to the Wilkes faculty or pharmacy faculty. And here is an example. It could be linking, okay, all these aptamer or DNA can be linked to some of the cytotoxic agents, okay, for example doxorubicins, and these are DNA intercalating agents. They can carry it, bind to a cancer cell as receptors, and hopefully it will internalize. Now here I also have another thing that I really didn't talk about at any point in time is something called near infrared frequencies or photothermal therapy. Basically is you put on a pretty harmless infrared near the cancer cells, okay, and these infrared will actually heat up the golden nanoparticle a little bit, leading the DNA to unfold and releasing the drug to kill the cancer cell. So it's almost science fiction in a way, but it actually can be done, at least in a dish format, in a petri dish. When you have cancer cell laid out inside the body, it's a little bit difficult, and like many other drugs or many other biologics inside the body, the stories are complicated. Right, in terms of infectious disease, okay, my point of view, so there are infectious disease. A lot of time I'm referring mostly on the bacterial infectious disease, okay, and they are the contributing factor for these bacterial diseases a lot of time because of their virulence factors or bacterial exotoxins. Okay, we kind of touch upon these exotoxins in the immunology chapter. So, but here don't worry about what what virulence factors we're talking about. But the idea here is aptamers can competitively bind to those toxins. And here is a kind of unrelated type of a picture and a paper that I pulled from an article saying that, hey, if you know, here it shows you a alpha toxins, okay, alpha toxins from Staph aureus, okay, if you know, the wound, okay, this is on a mice, okay, if it is infected with wild type Staph aureus, it will lead to you know, having these alpha toxin being released and lead to wound being delayed healing, okay, healed slower. If you're infected with a string of Staph aureus that cannot produce these virulence factors, you know, your wound really, you know, doesn't look too bad at all, okay, it just recovered. And anyway, so that is the recover much faster, or less, the lesion is much smaller. So the idea is, can we neutralize these toxins with these toxin specific aptamer, okay? So for example, an anti-alpha toxin aptamer. So I in the past I have identified one of those, and that helped me graduated, get my PhD. So now I didn't do for a therapeutic application, I did for diagnostic application. So so that's just a potential use, you know, I may revisit in my near future. So talking about diagnostics, when we are moving on to the diagnostic topic again, okay, so this paper it's published in 2015 with my advisor, Dr. Letha Suter, okay, my graduate advisor, Dr. Letha Suter. And this is a very, very long review paper, and some of you do research with me, I ask you to look at it, skim through it a little bit, but the P4s, okay, this is one of your required reading. You spend a whole day read it, okay? It's not that bad. The language are pretty comprehensible, you know, because I write it, you know, I don't write difficult stuff to read, you know, I cannot write difficult stuff, so it's pretty fluid, and you can read it very you know, in a reasonable time, I believe. So that is required for P4, not for anyone else that is watching this video again. All right, so in that article I highlight a couple things, and there are so many aptamers, okay, these days targeting pathogens, okay, infectious disease related materials, okay. It could be like a whole bacteria, okay, large increase since 2007. Before 2007 it hasn't, not that much, and at that point it just jumped. All right, so there's so many different, you know, these are the details you don't need to memorize in any way. You know, like I said, even if I ask on exam things in this lecture, and I won't ask you these number changes all the time, okay. So at one point in time there are just so many different aptamers or molecular recognition elements, okay, that are binding to different bacteria, whole cell bacteria, common one, E. coli, Salmonella, Listeria, Staph aureus, Streptococcus, you know, all these different type of bacteria. You name it, there could be it could be also endotoxins, some LPS, peptidoglycans, all these bacterial specific molecules, you know, are the target. Viruses, okay, a lot of research were done in HIV viral proteins. Unfortunately, really there's nothing fruitful coming out, you know, after all these time. That is an unfortunate thing. Influenza A viruses, okay, in terms of both therapeutic and detections, it's a big thing. And what are you thinking? Are you thinking what I'm thinking? A cold, okay. So we're gonna, you know, come back, you know, in a wrap up at the end of the lecture. That's why I tell you to read all the way, watch all the way to the end, even though this one is getting a little longer, but I hope my presentation is a little bit entertaining. Now it's getting to, you know, close to an hour mark. You know, take a break if you need to and come back, okay. So now that we, you know, there are also, it could be also virulence factors, right, like I pointed out earlier on, for the alpha toxins, okay, there are many different toxins in this world from bacteria. Cholera toxins it makes you diarrhea, you know, bacillus, you know, all these, you know, anthrax, okay, anthrax toxins, and bacillus anthracis, okay, that's, you know, the bacteria that is responsible for anthrax. Alright, cyanobacteria, some of those are marine, okay, marine organisms that secrete cyanobacteria can make if you can have like contaminated seafood, and if you eat those, you get poisoned by those fungal toxins, some of the mold toxins. These things are very big in subtropical weathered country, okay, like Southeast Asia. Like molding is a big thing, contaminating food like peanuts, and these are cancerous toxins. So detections of these cancerous materials are very, very crucial in many ways. Alright, so let's take one for example, toxin B. Okay, toxin B from where? From C. diff. Okay, toxin B is from C. diff. It accounts for many about 30% of all diarrheal cases in acute care setting, and it's costing millions and billions of dollars. Okay, per year in the U.S. alone, and it actually have an increasing mortality rate from this C. diff and infections, particularly in hospitalized patients. Okay, more recently in 2017 type of a data. Okay, there are so many deaths. Okay, don't worry about the number. I'm just trying to point out to you they are serious things. And a little bit background. For this disease to happen, you need to have a colonization of the toxic string of the C. diff, or like Clostridium difficile, and this particular protein, okay, inhibits member of some of the Rho family GTPase. Basically, it's causing cell dysfunction. Okay, that is the end story. Cell dysfunctions and current diagnosis. Okay, basically you need multiple diagnoses to confirm. Okay, enzyme immunoassays, like what I talked about, immunoassay ELISAs, things that we talked about in diagnostic chapter. Okay, ELISA, PCR, cell culture, pretty standard way of diagnostic. Right, so the idea here is like, hey, can we, or what I did was to find out a toxin B specific ssDNA aptamer or molecular recognition elements, okay, that can bind to this toxin in a fecal matter. So this is what I found again. So there's an example of aptamer, what potentially could look like in a 2D structure. In 3D, we have very little way of knowing it for sure. So it could be quite funny looking. Usually, like I said, there are still some of these stem loop and bulges or hairpin. You name it. Okay, there's these regions that they believe these regions are responsible for binding to the particular target. All right, viewer discretions. Okay. Alright, so this is what I did. You've seen it, okay? Some of you have seen it, at least the P2s have seen it, and this is the fecal sample preparations when I was a student. Okay, it is very disgusting, and I spiked the toxin B there. The idea is to develop some type of a ELISA-like assays. Okay, ELISA-like assays where you can detect these toxins in this complex matrices. Okay, ELISA assays, the key thing here, we have these horseradish peroxidase at the end. Okay, so that it catalyzes the reaction when there is the target present. You will have, the more you have, the more green or darker it will turn. So that you can visually tell a sample if there's more toxins versus less toxins. Now not every single component here is an antibody, so I swap out one of the antibody with my aptamer that is represented with this squiggly line here, squiggly blue line here. Alright, now you don't need to know the detail about how these things work. Just here, I'm just presenting you as a complete picture. Again, all these data, you don't need to know any, in a specific way. Here, I'm just showing you, hey, it was somewhat successful to detect these toxins in some type of a fecal solutions. Okay, so it was a difficult experiment. I sometimes tell my students, research students, that I did about 13-14 essays like that in the picture, and I got maybe 3 or 4 that was that actually work. Other diagnostic applications, okay, a lot of the time here, I like to focus on is the monitoring drug concentrations in plasma, particularly some of the antibiotics, anticoagulants, or things or drugs that with narrow therapeutic windows, can also be useful for bedside diagnostic, or maybe develop into some type of a lateral flow assays. Okay, lateral flow assay that we talked about. Essentially, you just swap out some of the antibody with aptamer in it. Now, okay, some of the real work, again, I want to credit my students, okay, that worked with me. All right, so we have Navina, P3, okay, Navina, she's been the excellent student in the lab. Everyone is good, but she's particularly more, skill and research oriented in many ways. And then she helped with a project that are identifying a DNA specific aptamer that is specific for a direct factor 10A inhibitor. Okay, so it is a drug. So it is not direct factor 10A, it's factor 10A inhibitor. So one of those XA bands, okay, that is, very common these days, right? One of the three, I won't disclose it yet. Okay, one of the three. And so I want to credit, as you see, Navina here and someone in your class, P2, Kirsten. Okay, Kirsten, she's been very helpful along the way as well. And other students that I will name them later on, will accredit them. So everyone here is student except me in this project. Now, okay, this project was originally going to present in the ACS conference in Philadelphia in March, but we all know that not everything canceled, right? COVID cancels everything except student loans, right? So that is something that I saw online. Anyhow, so we still get a chance to present this project poster digitally, okay, submit online. So that is thanks to all of students' hard work. Okay, thanks to all of students' hard work. Alright, now going to biosensors. Okay, biosensors, it's a little smaller topic. It is essentially, it's a off branch of diagnostic. Okay, when we talking about sensing health related material, we talk about as diagnostic. When we are sensing things that are not particularly disease related, we call it biosensing. Okay, and I have a book chapter published last year. Okay, it's about a year ago. Last year was a big year actually. I got a lot of things done. So this particular one, we're talking about some of the I was talking about the tools and biosensors that based on DNA and RNA, in particular these aptamer molecules, and how it used to monitor environmental toxins. Okay, environmental toxins. Again, this is the last article or chapter that the P4s will read for their epi experience, and P2s and everyone else, again, don't need to read this. Okay, it is not as long as the 2015 publication, but it goes into areas that are potentially go over your head. Actually, it went over my head too when I was writing it. I just had to learn it on the spot in terms of some of the engineering aspect of these sensors development. Alright, in a nutshell, highlights of major biosensing principles. I'm not going to talk about the mechanism of it. I don't think it's relevant enough. But basically, the category, we have something called like an electrochemical sensor, i.e. your things like diabetes monitoring machines. Okay, there is electronic components in it, and it can sense your glucose levels, right? Okay, that is one of those examples. And we can big category as optical. So optical, we talked about fluorescent energy transfer. Okay, when we talked about diagnostic, absorbance. Okay, so things like the light transmissions. Colorimetric, okay, gold nanoparticle. Again, we talked about the principles of that when we talked about lateral flow assays. Okay, now the big category in terms of biosensors or apter sensors, we call it apter sensors in the field of toxicology, environmental toxicology. About over half is based on fluorescent principles and colorimetric principles, and that's why I presented those two concepts to you guys when we talked about diagnostic. And the rest of it is just or electrochemicals and other mechanisms takes about a quarter to a total of about half of those aptasensor out there. All right. Okay. So what are the environmental toxins? And we are looking at chemical toxins usually. A lot of them are endocrine disruptors, estradiols coming off from a lot of the oral contraceptives, okay, being excreted back into the environment, and other hormonal replacement therapy, testosterone, estrogen, everything that could be leaked into the water, okay, water supplies and things like that. Bisphenol A, some of those are industrial products. Again, you don't need to know what these things do, just industrial product. They are either disrupt your endocrine systems or being cancerous in many cases. Herbicides, pesticides, one of those bigger thing. Okay, you apply it into the field, the rain wash it off, and goes into the to the river. Okay, now this river is in I took this picture in Annapolis, Maryland. Okay, anyone have been there? I don't know. I used to live there when I first came to the country 17 years ago. It seems very long time ago. This little guy, I took this picture. I took this picture one summer when I was growing some type of a pumpkin thing. I think in the background here, I think a pumpkin, just for fun. And I took this little grasshopper. Okay, so some of the work, again, these work are student work. That's why I'm accrediting them again. At the end, we'll accredit them. So the atrazine, okay, one of those atrazine herbicides called atrazine, and they are very widely used in the U.S. They are banned in Europe. Okay, actually they are banned in Europe, but U.S. doesn't seem to ban this product, and they are used in everywhere. Again, don't worry about the detail. Lawns and agricultural, and they are persistent in the environment. Actually, they have been shown to contaminate waters, including ground surface drinking waters. And if you have a refrigerator filter at home, like those GE brand refrigerator filter, if your household own one of those refrigerator come with a filter, the chances are when you buy new, there's some product information saying that it can filter so much atrazine, okay, because it is a real thing out there, okay. Has been linked to it's a non-hormonal disruptor, okay, also linked to birth defects and some form of a cancer as well. So it is a bad thing. Okay, and we don't want it in our drinking water. Now, the idea is we want to detect it too, right? If we cannot detect it precisely, how can we say this is a safe drinking water, right? So, what are the work that has been done in this area or in this particular topic? So, Kevin Abraham and Mina Rurenthal, they were the MS Bioengineering student, okay, that graduated, and they both have different each one have one article in this regard to look for an aptamer that is specific for atrazine and developing some type of a econometric sensing principle. So here is some of those graph showing you, hey, this, showing you for real. I always just say, when you detect something, it'll turn purple. When it doesn't detect something, it kind of stay kind of pinkish. You can see the differences both in a picture way and in a graphical data way, plotted out in a numerical way. And here picture shows you a gradual change in a dose-dependent manner. So these are all student works, and I think I cannot thank them enough. So this video is dedicated for all these student works, actually. All right. So more recently or currently, what's happening with my lab here? We are looking at another herbicide. It's called a picolinate in the picolinic acid group. So it's basically a carboxylic acid type of a herbicide. I won't disclose the herbicide name yet because it's not really published. So this particular herbicide can resist hydrolysis and photolysis and has been shown to contaminate compost pile, and particularly in the areas that are colder. Okay, this particular herbicide can only be degraded by bacteria, and if you're cold enough, bacteria is not active and these things stays there for a long time. And I have to thank again this group of students, and one of them here is Kenneth Wilkes. He is the current student that is still in the program that are expecting to graduate in May or finishing up. I know it's a tricky situation for everyone in the school. He's an MS bioengineering student and he's been working with me since last summer, actually since last June. He's been great on this project, and again, Navinas and Kirsten, they were also helping with this particular project as well. So it is a teamwork, group work, and I'm fortunate to have everyone here on the team. So that is the present and future. Now the future, let's go tie back to the broader theme that I've been talking, for these couple weeks, in terms of the COVID-19, right, SARS-CoV-2 diagnostics. Okay, so what I envisioned myself of doing in a very near future is something titled called DNA aptamer-mediated plasmonic detections of SARS-CoV-2. Whoa, that's a very fancy name. We always want to have some type of a fancy title, so that it'll catch people's eye. Right, a lot of time when you're writing, just a trivial thing when I write grants, and you want something that are eye catchy. You want something that not sounds boring. Okay, people say, what is this? Let me read into it. If you see something that are let's just like you're reading a book, right, or reading a article with a title. The title catch you to click into it to read, right? So that is the idea, to have a catchy title, in my opinion. Central idea to select a DNA aptamer against components of SARS-CoV-2 with high affinity and specificity. So kind of resonate back into what I talked about on Tuesday in terms of maybe we can find something that bind to the SB domain of the S1 subunit. Okay, so it could be stopping. Okay, it could be therapeutic in a sense, but my focus is more on diagnostic and biosensing. So that's why I don't move too much into the therapeutic standpoint at this point. So it could be the SB domain, or it could be the spike envelope membrane mosaic protein. So we have this CoV-2. Oh yeah, my drawing is terrible. Did I draw all these thumbnail? Yes, I draw all these thumbnail in the videos that you saw previously. The psoriasis, the liver, the bunny antibody, the angry antibody. I drew all those, but it took me a long time to draw those. So on the spot, I drew it very terrible with this same technology here. Now, so here is the S protein, right? S spike and the envelope. Okay. These are there you have empty space on the envelope, right? So those have protein as well, so we could target those proteins. Non-specifically, okay, if I could acquire some type of an inactivated whole bacteria that are non-virulent in nature, well, these things are difficult. I mean, I'm actually scared if I were to get like a whole virus. I don't know if I would be able to do it, honestly. If I were in a different facility with a better control, perhaps wearing like a N95 mask or P100 mask all the time, then the chances are, if you are screening against the whole virus, you can find novel targets that it can bind to. So there are some value to it. It's dangerous, high risk, high return type of a research. Applications wise, you can develop LFA, lateral flow assays, and also something called plasmonic enzyme linked oligonucleotide assays. Oh, very fancy name. Essentially it's just coupling gold nanoparticle with these ELISA like assays. That's all. So that is the that is the future that I believe that I see myself potentially doing, given enough funding and time, and again students really is the driving force everywhere. When I was a student, I was a driving force, and now I'm a teaching faculty here. The students again is the driving force, and I value every one of you, not only those in the lab, every one of you in class as well, because you are my audience. Okay, so finally we come to the end, wrapping up. It is almost an hour 15 minutes into it, and that's really all the content, and I would like to give them some acknowledgments. Now, if you pay enough attention of some of the authors that I quoted, that you may notice something, or you may have not noticed something if you fall asleep during halfway. Now, here is the acknowledgement of my academic pedigree. Yay! So I'm very proud that I am one of the descendants of Jack Szostak, Dr. Jack Szostak. Okay, here highlighted in the middle with the Nobel Prize medal. Now he has a mentor. It's called Ray Wu in Cornell University, and he actually was the postdoctoral advisor of Dr. Ellington, and Ellington is the graduate advisor of my advisors, Dr. Letha Suter, and I'm fortunately one of the two students she graduated, primarily graduated, that is out there. So I am lucky in a way that I'm 3 generations away, like 3 generations away from a Nobel Prize. That's as close as I could get, I feel like. But anyway, so I really acknowledge them. Without Dr. Szostak and Ellington and everyone above me, I could not be doing what I'm doing, and it is definitely an acknowledgement of academic in that sense. In the past, and I also want to definitely acknowledge my graduate advisor. So here is a better picture of me. I look a lot younger. I feel like these 5 years has aged me tremendously. This past month, I feel like has aged me tremendously. And this is me and my advisor, Letha Suter, back in 2015 May, in Morgantown, West Virginia, when I walked and got my hootings. Okay, and so in my dissertation, I was if in case you haven't noticed, maybe I've never told you guys, I am a Star Trek fan, and I in the Star Trek theme, they're always space, the final frontier, we are the voyage of the Starship Enterprise and things like that. So I adopted this little quote here: science, the final frontier. I almost treated it as a space adventure. To me, it is really a scientific adventure for me during the time when I was as a student in Suter Lab, and I acknowledged her in my dissertations in a very unique way, as I would say. And I'm fortunate enough now that I have a small crew of myself. In my free time, I imagined myself of doing some type of a scientific research that are out there that no one's are doing, but actually a lot of people are doing. So this type of things, I actually am writing some type of a creative essays in terms of describing my life. So one day you may see something published in that regard. Alright, I'll just hint that much. Okay, that's it. And the acknowledgement for present. Okay, how can I not acknowledge my crew here at Wilkes, right? So pharmacy school, here we have Dr. Alex Pons, and he just matched for another year of PG fellowship something. Okay, she's doing a fellowship in the starting in the summer, hopefully, with everything that are calming down. Okay, in the current graduating class, okay, Mia Lucia, Danica Benson, okay, P4, your fellow castmates, two of them. In the P3 class, Navina Chunso, in the P2 class. All right, so I'm finally exposing, who's doing research with me. Kirsten. Okay, Kirsten, this is something to be proud of. Okay, don't blush. Okay, don't blush. No one sees you. You're at home. Okay, and P1 in the P1 class we have Paige and Preform. Okay, I have a pharmacy Preform. Julia Isker. I don't have pictures of them. Okay, but they are helping in a way. Some of them have a little bit more, so there's a little bit growing thing, but still they contributed positively to some of the project in the that are currently in work. And the Wilkes Bioengineering side, I would like to thank Kevin Abraham, my first MS student here. Kevin here is this little Kevin here. Okay, Mina Rurenthor. Actually, she didn't get her MS with me, but she joined the lab as the research assistant for from the summer to the end, about 6-7 months, and she was very productive in many ways. So I acknowledge her, and my current MS student Kenneth Kaiser here with the glasses, and your beloved fellow classmate Lavina and Kirsten. Okay, so please don't mind that I showed a picture. The picture I showed is just to acknowledge everyone's contribution. I really want to thank everyone here that helped me along the way. Okay, without students there would be no lab activities. That's seriously how I think about it. Alright, trivia. So stay here. There's one more slide. Okay, one more slide. Don't go yet. Trivia. I highlighted 1990 last time. It's a special year. So it is a 30th year for many things. Okay, phage display library, nucleic acid aptamer, Cellax, and actually also a 30th year for commercialization of a special instrument called surface plasma resonance instrument that I used exclusively when I was a student, and also have a basic unit here in the Wilkes lab that I haven't used it very much at all, but that is just some of the things that is interesting to me. 1990 seems to be a very big year for many reasons for me. Now I wasn't born in 1990. Okay, so I'm over 30 years old. I'm just gonna tell you, in case you wonder, am I 30 years? I'm over. Okay, so last assignment. So this is for P2 only. Alright, this is for P2 only. So if you are your P4s, and just stick a little bit longer. I have an end message after this, and I want you to stick here to just watch in the entirety. But this assignment's for P2 only, so I'm going to let you read a newsletter. Okay, this is not a typical article, just a newsletter from the International Society on Aptamer. Okay, I ask you to read it because I have a small contribution to that newsletter. That's about my extent of involvement in the society. Actually, I am not the officers or anything, but there is a society on Aptamer out there. I want you guys to reflect on what new information you've learned from the article and some of the general topic on ligand evolutions. I believe most of the things that you're learning are new, so feel free to write your responses. Of course, be professional. Okay, there are no specific grading rubric for this assignment. Okay, I'm really just grading based on on-time submissions before the deadline that was set on D2L. Okay, 1 to 2 page, double-spaced. Remember, I always read every single one of them. Okay, sometimes my comments are a little bit shorter because I just have been reading so many of them, and I get to a point a little bit tired. And D2L submissions as usual. Alright, so that is the end of the material, and then my message is here. Alright guys, I hope the lecture was entertaining enough. I know there's some type of a small glitch in the audio. I think there was some type of a static went on when I was recording. I am very sorry about that if that bothers you. Usually there's a short period of went on with the recording. I think what happened was my cell phone was too close to my microphone, and the signal jammed into the recording, so I apologize for that. But I try to minimize it. I try to do a little bit editing to minimize that effect as much as possible. Alright, so now this semester, this is the last video that I am uploading, okay, in terms of my lecture. I hope you've all enjoyed enough. I try to do multiple things for you guys, and especially for this biotechnology topic, I built it almost like try to be like a small web series. Okay, what it means is that there is a individual lecture where it is self-contained with a story behind it, and then if you look at the entire biotechnology arc per se, there is some type of a tied into it. You keep seeing me, or you keep listening to me referring to some material that was previously presented, such as the PCR lateral flow assays and some of the recombinant DNA technology, and all these, they all kind of sync into together. And there is a bigger theme that I built in that was not planned, especially in January, was the COVID-19 or SARS-CoV-2 topic. So everything is brand new here, and I hope that I've presented good enough information that you've learned something, and I know this is a difficult time now, and for the P4s, I know this is very difficult, and I hope that my lecture give you a background good enough that will help you move on into the reading assignments as well. Okay, so that is my overall message, ending message with you all, and best wish with your other finals, P2s, and your final exam message will be up at 9 o'clock on Friday. It is all written up, scheduled. Okay, so that announcement will be posted automatically at that time. Okay, so that is really the end of my voice. Now you will see my writing in my response, but that is the end of my voice, and I am honored to have this chance to teach you all. Although we're not face to face, I don't see you, but you do see me. Okay, alright, so that's that, and I will see you virtually, kind of see your writing on exams and assignments, and I will see you in the fall semester. Bye.